besides the speakers, we'd like to welcome David Guterman from the Medical College of Wisconsin and Stephen Macknick from the SUNY Downstate to the panel. And I guess to follow what was done in the first panel, we'll have Dr. Guterman and Dr. Macknick say a few words to start out.
Okay, great. Thank you. All right. Well, I have a whole lot of comments I could make, but I'll just start out with a few because I think the discussion from the audience is really quite helpful. Following up on one of the items that Dr. Chilian talked about has to do with the cardiomyocytes producing hydrogen peroxide that then interact in the smooth muscle. And John Canty published a number of years ago editorial talking about hydrogen peroxide kind of attacking the blood vessel or the smooth muscle from two different angles. And some of the work that we've done and others have shown that the endothelial cells also produce hydrogen peroxide as a vascular vasodilator mechanism for flow-mediated dilation. So you have the endothelial cells as well as the cardiac myocytes in the heart impinging on the smooth muscle cells with hydrogen peroxide. But the point I wanted to make is that, as Dr. Chilian mentioned, probably the metabolic dilation produced by this hydrogen peroxide is a transient phenomenon, and hydrogen peroxide levels go up and down. From the endothelial cells, what we see, at least in patients with coronary disease, this release of hydrogen peroxide seems to be fairly continuous in isolated vessels from the endothelium. So it may actually invoke some pathological changes depending on what's being released from the endothelial cell. If you have a normal patient, what is being released from this huge array of microcirculatory vessels in the heart is nitric oxide. But as soon as a patient develops coronary disease, there's a switch to hydrogen peroxide. So part of the pathology that we see, and we know that microvessel disease occurs in humans, we measure it very roughly or crudely by just measuring an alteration in maximum increase in blood flow with adenosine or other dilators. But we know that this abnormality occurs, has just as bad a prognosis or worse than macrovascular disease does, but we don't understand how that's occurring, and we need to really look more specifically at microvessel function, I think, to understand that. We need better techniques in the patients to be able to understand what's going on at the microvascular level. So that's just one example, I think, where hydrogen peroxide may be a bad player, nitric oxide a good player, and the microcirculation being the producer of these elements that's in bathing parenchymal cells, whether it be heart or elsewhere. So I think this is one of the big gaps in our knowledge with that, in regard to that issue. I also wanted to mention on Jerry Meininger's presentation talking about the heterogeneity of vascular smooth muscles, even in the same microvessel that's pulled out of a tissue. We see, and I think others have also published, that the content of potassium channels expressed in individual smooth muscle cells dispersed or isolated from fresh tissue samples may vary quite greatly. I'm not sure what the meaning of that is or what the purpose is, but there's quite a bit of heterogeneity just in the expression of various surface proteins in smooth muscle cells from adjacent, presumably adjacent cells and adjacent tissue. I can stop there.
Well, my comments and questions lead largely from Kleinfeld's talk, which I think is a really important anchor for this meeting, really, in terms of the basic neuroscience there, at least in the brain. And I think probably the most important concept that he brought up was this issue of coalescence. So you have one penetrating arterial blockage that leads to a massive behavioral change that can be reversed and shown in, with psychophysics of a whole animal to affect these large behavioral improvements. And I think that one of the things that or rather deficits, and one of the things that suggests is that these small vessel diseases can be the 1,000 little pinpricks that lead to humongous changes. Now, one of the things that I was interested in was the fact that a single penetrating arterial blockage, even though the capillary beds are connected to the surrounding penetrating arterial capillary beds, that the pressure wasn't high enough to allow the blood flow to cross or to feed the cells in there. And I wonder if blood flow enhancers would decrease the size of the umbra from a single arterial blockage, penetrating arterial blockage in that case.
So, all right. Your question was why, if you increase flow, like you've gotten blood into this region that was.
Well, it was more if you increase blood flow with nitric oxide, for example.
Oh.
If you increase blood flow with nitric oxide.
This is a new building.
So you
I
used
think it's years of teaching physics 1 where you got to shout.
so you've used excitotoxic
Right.
drugs.
We went down a lot of blind. I mean, the truth is, we went down a huge number of blind alleys looking for inflammation. We didn't actually increase pressure. I mean, it's a fair point. It should have been done. I mean, I think though the point is that it's not. The real issue is that the venules have very little resistance, so they should just be sopping up the blood. I mean, if there's actually a way. I mean, the prediction, if it's possible, would be if there's a way to actually constrict the local veins. Now, I realize they don't have smooth muscle, but if there's any trick out there, right, to constrict the veins, okay, then in fact blood should get in from the side.
I see. So it's not the arteriolar side that's going to get the umbra smaller. It's the venule side.
It's the venule side. It's the drains, and it's just maybe it's worth thinking about. I really have no clue how to do this.
Okay. And my other comment was for Chilean, which was that so you showed linear relationships between blood flow and metabolism, and are those two ways? So, I mean, do you have effects on metabolism that leads to changes in blood flow, and will the changes in blood flow happening for whatever reason lead to the same correlated changes in metabolism, or is it really causal only from the metabolism to the blood flow?
In the heart, there's a phenomenon called the Gregg effect, and that is if you increase blood flow and increase the vascular volume of the myocardium, you increase the work that it's done. And it's probably due to the contraction of the muscle. If you have more water in the muscle, so to speak, it takes more energy to overcome and move that mass.
So we can take some questions from the floor.
Connie Wyand from Stanford to Dr. Chilean. Bill, so if H2O2 is very important in regulating this, the immune cells are really born and equipped to make H2O2. They make it better than any other cell. Do they regulate blood flow in the heart?
I don't know. I think that's an intriguing idea because certainly the myocardium has an ability to recruit many types of immune cells under a variety of conditions, even conditions that are not profound frank ischemia, but it would just be, or there could some ischemic episodes, but not enough to cause necrosis. And clearly that could be a mechanism.
Yeah, this is a comment for David Kleinfeld. So you mentioned that the bottleneck is at the penetrating arterial. We've done experiments in which we embolize vessels of any size, including like a 10 micron terminal arterial, I guess, and we see a tiny microcore of cell death there.
Yeah.
So basically, I think that even the smallest vessels are actually there's not enough collateral circulation to compensate for those occlusions.
Okay, so 10 microns is I mean, these would be, what would you call them? Sort of like primary capillaries, things coming off the penetrating vessels. Mm-hmm. Would that be a fair description?
So they would be deep into the brain.
Deep into the brain,
deep in the brain, branches of.
But big enough to supply multiple
maybe
I'm trying to get a hierarchy.
like a two capillary. Basically, it's pre-branch, you know. Sort of
Pre-branch.
yeah.
So maybe, okay, you sort of, it's good to put a word on something because some people remember it, but maybe a fairer way to say it is you block a penetrating vessel, you get a quite extended amount of death. In our hands, it's, this half millimeter. When you go down to just to a very small vessel in the mouse, like things that are like 3-4, do you see a cyst?
You don't see a cyst, but if you do high quality histology, you will see that there is a microcore, maybe a couple of dead cells with gliosis and a clear reaction, basically, pathological reaction.
Okay, so I have to say that when we blocked the smallest microvessels, we didn't really see death, but that, I don't think it's very important. A few neurons, what's a few neurons? No, but in a serious way, so maybe there's a progression, but I don't think you get this large-scale cyst. I think maybe that's consistent when you block a very small vessel, and I don't, it may not be a threshold phenomena, right?
Howard Crystal, SUNY Downstate. This is for Dr. Kleinfeld also. Could you comment on the human pathologic correlates, in particular because clinicians are always talking about white matter. To what extent are your findings because it's an artifact that you can study the cortex more easily? And why don't we see these cysts in postmortem material?
Okay, well, I understand. I wasn't trained in medicine, so and I try to avoid the physicians too. But I think what you do see, what you see in Harry Vinter's published pathology data, and now what you see in the 7 Tesla data coming from George Biesel's lab, but it's only the last year and a half, are gray matter lesions. Now, the criticism perhaps is that the 7 Tesla work is still at the level of MRI. These patients are still alive, so there's not a correlation with postmortem tissue. So I think it was a technological reason that people didn't see these. It's only when you have 7 Tesla that you get below about 1 millimeter resolution, but it was already there in the pathology data. So it's just, it's not a proof. You look at a mouse model, or you block one vessel, you see a certain size defect. It's water rich. T2 relaxation goes up. You look in the human recent human data, you see the same kind of signature. It may take a couple of years to match. I think the reason you saw this in white matter, white matter is very water poor. Okay, so the contrast levels were much higher. Okay, and then it raises another interesting question: is why also the defects tended to spread over a larger region. I think that's going to be a challenge. That's part of the challenges, right, for mouse models. But I have a feeling it wasn't that it wasn't there. It just wasn't seen as a technological issue of resolution in the magnets.
Thank you.
Okay. And so if we could have kind of short answers and short questions so we could get all the people that are currently standing and nobody else. Thank you.
John Lederer, University of Maryland, for Dr. Chilean, but also I was hoping the rest of the panel would have perhaps a comment or a thought. In heart, when heart cells are stretched, they have a physiologic generation of ROS in the form, we think, of hydrogen peroxide that produces local signals, but it goes through superoxide outside the cell. Given the intimate nature of the capillaries to the heart cell, 1-to-1, real close, there should be a physiologic ROS source with every heartbeat that should also have an effect. Can you, first of all, consider that? And secondly, speculate, could that account in part for some of the ROS effects that you may be seeing for vasodilation?
I certainly cannot eliminate it. If one would stretch cardiac myocytes and you move up on the so-called Frank-Starling relationship and the heart's doing more work, it's going to produce more hydrogen peroxide.
You get 8% with every filling during every heartbeat, and that's enough to produce a signal.
An 8% change in
8%.
Hydrogen peroxide?
No, of stretch.
Oh, okay. But in terms of beat-to-beat regulation, my hunch would be yes. It must play a role. I think the work that comes to my mind, this is old work, but it was work done by Joseph C. Greenfield many years ago showing that even a slight occlusion during a portion of diastole resulted in a compensatory hyperemic response. So it can happen very quickly.
So I don't know if anyone else has a thought on hydrogen peroxide in the systems.
So I think that's absolutely right. And if you look at the peripheral vasculature, even outside the heart, just the pulsatility, Gabor Cayley and others in Valhalla have shown that just stretching the vessel and others produce superoxide and hydrogen peroxide. So this probably effect goes beyond the heart into the peripheral vasculature. And a clinical situation might be with the most recent onset of use of LVADs, which now produce a more continuous flow rather than a pulsatile flow in the periphery, and may induce some abnormalities in relation to that.
Manfred Böhm, Lange Blood Institute. I have a question for Dr. Meininger. Response to injury theory or dogma. There is the switch of contractile smooth muscle cell to a proliferative secretory smooth muscle cell. There is a very rare disease where there's a variant, actually a loss of function variant in soluble granulate cyclase, which is a receptor for NO. The hypothesis would be that these smooth muscle cells have a
Just try to speak more in the microphone.
So, classic response to injury, smooth muscle cells switch.
Even closer.
Even closer?
Even closer.
Okay. Perfect. In the classical response to injury, smooth muscle cells switch from a contractile phenotype to a proliferative phenotype, secretory phenotype. There's a rare disease where there's a defect in the contractility in smooth muscle cell that does not necessarily respond in a problem of hypertension or regulation of blood pressure. It's a response, a hyperproliferative response. So these patients develop moya or achalasia of the stomach. So my question is, if you have a defect in the contractile ability of a smooth muscle cell, is there a kind of switch to a proliferative smooth muscle cell, very similar to the classic theory, and how would that be regulated?
That's a complicated question. And it's difficult to give you a short answer to that. All of our observations in single cells are done in cells that are in primary culture, so they haven't been passaged. But they still change a little bit. We can't help that, but it's the nature of the technology we use to study them. They still do retain some contractile properties. So they're not entirely just proliferative. If we do a concentration response curve with angiotensin II, you can map the height of the cell, and you can see that the height increases with concentration. So they still retain some contractile properties. So I don't think that the responses I'm seeing are strictly because the cells have switched to a proliferative phenotype. It would be interesting to take cells that have switched and see if they're different. But we haven't done that.
So I'm sorry, I'm getting the signals from the bosses here. I'm sorry if you can find the people you wanted to speak to at lunchtime. So I want to thank everybody, the speakers and the panelists.